Sanai Simpson is a professor and department head at Purdue University with a Bachelor of Science degree in chemistry in 1998, master's degree in biochemistry from Gebze Technical University in 2000 in Turkey, and PhD in food science from Purdue University in 2006. After 15 years working in the plant science department at North Dakota State University, she has moved to her current position as department head, professor, and dean's chair in food science at Purdue University in July 2021. Dr. Sinse is a serial food chemist, and her research spans plant science, food science, and analytical chemistry. She has developed a research program on the integration of cereal quality with the structure-function relationship of carbohydrates. Her program focuses on value-adding strategies for cereals and their byproducts. The goal of her group is to increase starch and non-starch polysaccharides. To achieve this goal, Dr. Simpson has aimed to integrate partnerships between producers, scientists, and food processors to optimize research potential and develop novel cereal carbohydrate derived functional ingredients. She has authored and co-authored numerous research papers and conference contributions in the area of grain quality and carbohydrate chemistry. She has an active member of the Institute of Food Technologies, Cereal and Grain Association, and Wheat Quality Council. The first North Dakota State University Bear L. D'Apollonia Endowed Associate Professor in Cereal Science and Technology was awarded to Dr. Simpson in August 2013. She has been awarded the International Young Scientist Research Award in 2013 and the International Excellence in Teaching Award in 2019 by the Cereal and Grain Association. In 2020, Dr. Simpson was awarded the Anderson Cereals and Oilseeds Award of Excellence by NC 2013 U.S. Quality Grains Research Consortium. Simpson is an inspirational teacher and advisor, and her deep understanding of industries and challenges around the world has created successful research partnerships at a global scale. Thank you very much, Dr. Simpson, for participating in this cycle of seminars of the All Research Center here in Yautepec, Morelos, that you visited some years ago. Thank you very much.
Mm-hmm. Yes, Luis, I was going to say the same thing. Thank you so much for inviting me for today's seminar. And in the year of 2012, actually, 10 years ago, I visited your campus. It was great. So it is my honor to give this presentation today to your center. And the title of my presentation is Wheat Arabinos Islands and their insight into the structure and functionality. So I had some introductory slides about myself, but I'm going to pass that. So as he mentioned, my background is chemistry, PhD in food chemistry. And currently I am the head of the food science department. I am also an active member of the Whistler Center for Carbohydrates Research. Some of you know that Dr. Bello Perez also spent some time at Purdue. As he mentioned, my background is on carbohydrate chemistry, and my vision for carbohydrate chemistry research. So I will just say overall vision, because we have some projects with Arabinos Islands, starch, some other non-starch polysaccharides, beta-glucan, modified starch. But my vision has been always how we can look at the fine structure of carbohydrates at very basic level in terms of molecular weight, linkage analysis, degree of substitution, and how these properties are impacting rheological properties or viscosity properties, the functional properties. and then that function, how that is linked into the end-use applications. And recently, and you will see some of examples of my research projects, we take it into the next level, how those properties can actually impact biological functionality when they are consumed by the human through the different food products. And these are, again, just a summary slide of some of the examples of my projects. And the goal is bringing together fine structure, functionality, value adding into the cereal industry, and then also how those byproducts could be utilized in different types of applications. So I have some projects around metabolic profiling or encapsulation of phenolic compounds in the starch matrix. Again, some computer modeling studies, how carbohydrates are forming complexes. Sorry, so there is a comment about screen has 3 rectangles that block some parts of her presentation. Might be my Zoom screen. Do you still have the same issue, or? So I just completely turn off I do not see any presenters or anyone else. Do you think the problem is? Okay, I hope everything is okay. If you still have problem, please let me know. Luis, does everything look okay? Okay, the problem is still present. Yeah, he says I have there are some problems that it blocks the screen. I will try to share again. Okay, I guess we will just continue from here, but I hope it still is, you can see the screen. So we have also some projects on computer modeling studies, how carbohydrates are interacting with some small molecular weight compounds to form complexes, again, their biological activities, and also some of the biodegradable film formation studies. But today's topic is about arabinose islands, and more specific to wheat arabinose islands. And I also use the analogy of 360-degree approach because I looked at arabinose islands from different perspective, working with plant breeders. Their goal is to develop new varieties that have resistance to disease, they have high yield, and they have good functionality. But the arabinose islands, being part of the cell wall, they have important functionality for them. But then the next step is how arabinose islands are going to impact milling and flour and rheological properties, and then how they are going to impact end product quality once it's on the table, and after they are consumed by human, what is the effect of arabinose islands into the human health and nutrition, because we are considering arabinose islands as dietary fibers. And again, we can see this as a holistic view of arabinose islands or 360-degree approach. So let's talk about as kernel. So as you know, wheat is one of the most, actually the second cereal crop on earth that is consumed wide range of countries, wide range of different geographical regions. It has been raised for many thousands of years. And there are lots of studies about cell wall chemistry and arabinose island structure of wheat. And these are some of the major cereal crops and their total arabinose island percentages. So for wheat, the total arabinose island is ranging from 5.8 to 6%. So each cereal has major non-starch polysaccharide. For example, for oat, the major non-starch polysaccharide is beta-glucan. There is also a very high level of beta-glucan in barley, but for wheat and rye and triticale, the major non-starch polysaccharide is arabinoxylan. And arabinoxylan, and we have three botanical sections. We have three, but botanically we have three main compartments in wheat kernel. It's the embryo, endosperm, and bran. So embryo is inside the wheat kernel that we are using to extract refined white flour. You all know during the milling process. And then embryo is the part that plant reproduce itself. And then the bran is outer layer, which is also made of 7 other layers. So this bran is very rich in arabinoxylans. And aileron layer is the last layer of bran. That's actually it's still part of the layer between the bran and endosperm that is also very rich in arabinoxylans. And during the milling process, we are trying to remove aileron layer also. We just want to get endosperm as much as we can, but we are removing the aileron layer because aileron layer does not have gluten forming proteins. It is very rich in non-starch polysaccharides and vitamins and minerals. So these are some closer pictures of wheat kernels. This is the endosperm, aileron layer, and bran. And do we have arabinoxylans in the endosperm? Yes, but their levels are from 1 to 3%, much, much lower compared to other parts of the grain. So this is the structure of arabinoxylan. It is very similar to cellulosic structure. Why? Because it has beta-1,4 linkage, but in the industry, sometimes arabinoxylans are referred as pentoxans. Why? Because they are made of 5 carbon sugars. And so they are made of 5 carbon sugars, and the backbone is beta-1,4 linked xylose, and then it can have substitution in the 2 position or the 3 position. So this is the xylose backbone. Again, there is no substitution, but some of the xylose residues, they can have substitution in the 2 with arabinose, or it can have substitution in the 3 position with arabinose. And some of the arabinose residues, they can have ferulic acid. And the ferulic acids are very important because this ferulic acid can have another link into another arabinoxylan. Like that, you can imagine there is another parallel arabinoxylan molecule here. So they will be cross-linked to each other via ferulic acid linkage. So ferulic acid is very important for cross-linking. Why? We are going to talk about what type of arabinoxylans we can have in the cereal structures. We have water unextractable, which are not soluble in water, and then we have water extractable arabinoxylans. What is the difference is the cross-linking and the size of the molecule in terms of molecular weight. And there are also some other differences, but the molecular weight and the cross-linking, if we have high levels of them, they can make the water arabinoxylans unextractable in water systems. So I think it is important for us to understand the structure of arabinoxylans. Then some of rest of the presentation is in terms of their functionality and then also how we can study these details. One other thing I should also mention that if we have no arabinose substitution, I call this unsubstituted xylose residues. And then you can have one substitute, so it's monosubstitute xylose. In some of the arabinoxylans, some of the xylose residues, they can have substitution in the both 2 or the 3 position. Then you can have disubstitute arabinoxylan. Why it is important? It's important because the fine chemistry is impacting, again, rheological properties And also we have some studies showing that those details can also impact biological functionalities. So this is a small picture on how arabinose islands are deposited during the grain development. Wheat kernel development can be divided into the 3 stages. One is the completion of cellularization, and then the beginning of cell differentiation, and then also the beginning of maturation process. So arabinose islands are deposited in cell wall through vesicles, and then they merge with plasma membrane, and then first appear at cell differentiation stage. So in the second stage, and then they have the continuous and spatial and temporal changes during the maturation step. So we have done some studies on what controls the Arab. Now we know so far that what is the structure of arabinose islands, and then how we classify arabinose islands based on solubility, water extractable and unextractable. And then we talk super briefly about how they are produced or synthesized during the grain development stage. And what impacts the production of arabinose islands and the chemistry. We all know that we have different wheat classes, winter wheat, spring wheat, right? And then those classes are based on color. It could be white wheat, red wheat. We can have also based on hardness, hard wheat or soft wheat. So all these parameters are impacting the market class of wheat. And then within each wheat class, we have different wheat varieties. They have different parents. And then those wheat varieties are grown in different environments. They can have drought, heat, and some environments, the farmers are using irrigation. Sometimes it's natural. Like in California, wheat is raised with irrigation systems. And there is what impacts the most arabinose island production. This is not a simple response. Again, there are some studies that they actually, or the conclusion is environment has more impact on the total arabinose island, and then also arabinose to xylose ratios in different wheat classes, wheat varieties. In some studies, environment has more impact. And also how genotype and environment are interacting with each other. So again, there is high level of complexity in terms of total arabinose island, water extractable arabinose island, and then environment or genetics, which one is more important. So there are different studies. So this is one of our earlier studies that we checked first the arabinose island content in different environments and different varieties. So in the refined flour, which is coming from endosperm, or you can call that also white flour, arabinose island was ranging from 1.27 to 1.76%. There are some studies that they have up to 3%. Again, this is something also impacted by living conditions too. And there were also the variation in the varieties, arabinose to xylose ratio. Remember, we have the xylose backbone, and then you can have substitution, but not every xylose has substitution. So when we study arabinose island chemistry, we always check arabinose to xylose ratio, because this is giving us an indication of how much substitution we have. And then I will also show you how we study fine chemistry of arabinose islands with proton NMR2. So now let's look at milling and flour preparation. What are roles of arabinose islands? Again, as I mentioned a couple minutes ago, this is a cartoon version of wheat kernel. We have wheat bran, endosperm is the white area, and then the germ is here. So the total arabinose island in wheat bran is very high. It could be even up to 40%. There are some studies. And arabinose to xylose ratio is ranging from 0.8 to 1%. And then in whole wheat flour, this is or another study that arabinose, like whole wheat is when we grind the entire kernel, and you have the all anatomical parts of the grain, bran, germ, and embryo. And you can see how total arabinose island is ranging from 4% to 8 almost. In the refined flour, the highest is 2.68%. And then we also have variation in the arabinose to xylose structures. So what it tells us that arabinose islands, they have we have more of them in the bran, and then the chemistry of arabinose island is different in terms of in their location in the endosperm versus the bran. So we also check arabinose island content in Buehler mill streams. This is an experimental Buehler mill that we have 6 actually mill streams, 3 from the break and 3 from the reduction rolls. The goal of break rolls is to separate endosperm from bran and germ. And then the reduction is we are actually reducing the particle size. If you think about this picture here, during the Buehler mill is, again, we have 6 streams, but at commercial scale, there are hundreds of mill streams. And then each mill stream can have different flour from different parts of the grain. Some mill streams are going to have flours that are closer to outside layer, and some they are coming from the center of the kernel. So they can actually have differences in terms of their arabinose island content based on where is the location of that flour within the kernel. So we also see differences in arabinose island content. If they are closer to the bran layer, there is more arabinose island content. And the straight is when we combine all those streams. So this is the last part. This is just one example, and I am going to come back to this study and just one example that how fine chemistry can impact biological functionality. So I have I have some examples of arabinose islands, genetic impact, what happens during the milling process, some health benefits, and then I'm going to expand some of the topics and more specific examples of each area. So this is the cartoon version of arabinose island structure, right? We have the beta 1,4-link xylose residues, and there are different types of xylanases that they have different enzyme specificity. They have different they have different enzymatic action on the structure. So there is one xylanase, we call it GH11 family. It hydrolyzes xylose, arabinose island backbone when we have monosubstitute xylose residues. So this enzyme is hydrolyzing the backbone when you have next to the monosubstitute xylose residues. And then another enzyme is, so this is the blue one, it is hydrolyzing from the xylose backbone that is next to the monosubstitute xylose residue. The red one is the GH11. So blue is GH10, and then the red one is GH11, and it is hydrolyzing the backbone when we have in the unsubstitute xylose residues. And then we also have arabinofuracetases. They also have different enzyme activities. And alpha-L arabinofuracetate, that is the GH51 family, it is hydrolyzing the arabinose from monosubstitute xylose residues. And then the other one, the last one, is hydrolyzing from disubstitute xylose residues. So in this study, we treated arabinose islands in different combination of these enzymes, and we created different arabinose island hydrolysates that they are structurally tailored. And then we studied the chemical properties in terms of molecular weight, degree of substitution, and And I'm going to show some of those results. But then there was, again, we call them series, different families of arabinoxylene hydrolysates. So ANX families, CJX, BAF, CAF, those are the enzymes nomenclatures. So they have structurally distinct properties, and they had significant variation in the fine structure, fine chemistry. And then different enzymes, they actually produce different types of arabinoxylenes when we treat the substrate at different time and different order of these enzymes. So this study was important because then first we characterize those arabinoxylene hydrolysates and then check their functionalities. And I will come back to that. Now, end products, how arabinoxylenes are impacting the end product quality of cereal-based products. Arabinoxylenes are extremely water-loving molecules. They are very hydrophilic. That's why it is going to be important when you add water into the refined flour. There's a competition between the proteins and other carbohydrates and damaged starch, especially, how they are going to have interactions with water. So arabinoxylenes are very hydrophilic, and actually they can hold up to 10 times of their weight of water in those systems. So because they have strong interactions with water, they impact textural properties, shelf life, water binding capacity, stability of those samples. Cryostabilization is extremely important for frozen law applications, viscosity, and intermolecular interactions. I do not know if some of you have heard about oxidative gelation. Oxidative gelation is a mechanism that arabinoxylenes are forming a three-dimensional network that is under, if we have the hydrogen peroxide in the system. And if you call a system gel, that means you have a three-dimensional network that water is entrapped into the system. So arabinoxylenes are also important for oxidative gelation mechanisms in dough system. Again, this is a process that we have in the dough formation, especially in the bread baking process or pancakes. That system is extremely important to viscoelastic properties of dough. So we have arabinoxylenes in the dough system, but do they have positive impact on the gluten and starch network, or what is the impact of arabinoxylenes on bread quality? So these are confocal laser scanning microscope images of different dough samples, and they were actually added low molecular weight, medium molecular weight, and high molecular weight arabinoxylenes into the system. So here we know that starch granules are shown in green, and then the gluten is shown in red, and the arabinoxylenes, based on their molecular weight, they have different function in the dough system. In the normal gluten network, the starch and the gluten, they have interactions and they have a continuous phase, and the starch granules are embedded into the dough system. But if we have high molecular weight arabinoxylenes, they can actually impact that network. So here, we noticed that low molecular weight arabinoxylenes, they had positive impact on the gluten and the starch network. So again, since arabinoxylenes, as I mentioned you, that they have high water holding capacity. So we check water absorption of flour samples when we add small percentages of arabinoxylenes into the formulation. So in this case, we measure the water absorption with farinograph. Farinograph is, I'm sure you all know that it's an equipment to measure the dose stability, mixing properties, and then the absolute water absorption for flour samples. Because each flour, it requires optimum amount of water for optimum gluten development. So here, if we increase, we at 0.5% Arabinos Island, there was significant improvement in the water absorption. With the 1.5%, we see significant impact of water absorption. Again, it jumped from almost 58% to 64%. And then there was also impact on the stability. What stability is we present the stability in minutes, the farinograph stability. It tells us how long we can mix the dough until the gluten network breaks down. So also you can see here that the gluten stability was increased with increased levels of Arabinos Islands, but the major impact is on water absorption. Okay. So what Arabinos Islands, so that example shows that they have significant impact on the water absorption. Sometimes how much water that Arabinos Island holds, it can also impact some of the unique products in the baking industry. So refrigerated dough is one of the most popular products in the baking industry because the dough is stored at refrigerated conditions, and then when somebody wants to bake, you can just take out from the fridge, bake in 15-20 minutes, and you have freshly baked products. But one of the challenges with refrigerated dough is we have done series of studies on refrigerated dough is the Arabinos Islands in the flour, because they have high water holding capacity and refrigerated conditions. Even though they are cooler, there is still some enzymatic action. And then and we know that each flour, we have endogenous xylanase activity. The xylanase is the enzyme hydrolyzing Arabinos Island. So if Arabinos Islands are hydrolyzed, even at very, very low level, they will be releasing water into the dough system. And extra water in the dough system is impacting the protein chemistry, viscoelastic properties of dough. So that's why Arabinos Islands also have important functionality in refrigerated dough systems. So they can impact during storage. We have dough is losing the consistency and strength, and then Arabinos Islands becoming more soluble. There is some changes in the Arabinos Island chemistry in terms of Arabinose to xylose ratio and molecular weight. Again, we have done series of studies on what happens to proteins, starch, and how we can actually stabilize Arabinos Islands in refrigerated dough systems. So this is a cartoon, and this has been very early studies, but I still really like because this is a good summary of how Arabinos Islands are impacting the quality of bread during fermentation process. So here we are showing the water unextractable Arabinos Islands that are large in size with green ropes, and then water extractable Arabinos Islands are in the blue. So in a long story short, water extractable Arabinos Islands, which are smaller in size, they actually stabilize the gas cells that we are producing during dough fermentation process. So they are good for baking quality. And but water unextracted, this is the summary. If there is a question about if Arabinos Islands, what are the impact of Arabinos Islands in bread? Water extractable Arabinos Islands here, they increase stability of gas cells, they stabilize the gas again, and they improve the texture because they have low molecular weight. But the unextractable Arabinos Islands, they have lots of cross-linking, very large in size. They will disrupt the gluten matrix and they will lower the loaf volumes. So that's one of the reasons if enzymes are heavily used in the baking industry, or sometimes bakers talk about enzyme cocktails. Why? Because those xylanases with unique enzyme specificities, they can have significant applications in the baking industry to improve dough quality, handling properties, oven spring, loaf volume, and then also in the final products. So for bread, we know what are the roles of arabinose islands. We want them on a water extractable ones at low molecular weight. But with cookies, it's not desirable in the salt wheat cookies because because remember for bread, high water absorption is desirable. But for the cookie is low moisture food. We do not want to have high water absorption in the cookie system. So arabinose islands, they cause higher water absorption, longer bake time, and then poor cookie spread. So because they just make the doughs like thicker but not wide enough. Sorry, cookies very thick but not wide enough. That's why they are not desirable. So we have done again some studies on how arabinose islands are impacting dough rheological properties and how the fine chemistry is changing during baking process. So we use different varieties that we know they have differences in terms of their baking quality plus some dough rheological properties, but they are also commonly used by growers. So if you are eating wheat from a pizza dough, most of the pizza dough in North America, they have spring wheat in the formulations. So here we can actually see this is the amount of water extractable arabinose islands in the flour, and then we collected samples at mixing stage and then through the fermentation process and even in the final loaf of bread. So we monitor water extractable arabinose islands and unextractable arabinose islands, how the ratio is changing for both steps, for all the varieties. So as you can see, they have completely opposite trend. Why? Because some of the water unextractable arabinose islands are converted to water extractable arabinose islands because their content is decreasing from flour to bread, but then there is some increase. So they have completely opposite trend. And there are some changes in terms of solubility and then fine chemistry. And we studied the fine chemistry of arabinose islands using proton NMR. And this is just the representative proton NMR spectra that how we can actually assign. So this will be monosubstitute xylose, not unsubstitute xylose. With like here, unique peak for unsubstitute xylose. And then for monosubstitute, we have unique xylose peaks here. And then for disubstitute, they have this region. And then for, we also had arabinogalactones, and arabinose residues are also studied in using the proton NMR technology again. So we studied all these changes, and I did not show you all the details, but then we propose a mechanism, what happens to arabinose island during the dough mixing stage. So first, we found that water extractable arabinose island has higher arabinose substitution rate. And then second, the amount of monosubstitute xylose decreased based on the NMR results, and then amount of unsubstitute xylose molecules increase. So that monosubstitute xylose can be hydrolyzed to produce unsubstitute xylose. And then, since the amount of water on extractable arabinose island decreased, the amount of water extractable arabinose island is actually increased. This indicates that part of the unextractable arabinose islands, they were solubilized and became water extractable. And some of them, again, via solubilization, they moved into the water extractable region. And then when we put together all the molecular weight and then all the NMR data, we were able to develop a model that how the fine chemistry was changing. So these are again some studies of arabinose islands on how they are changing during the bread baking process. But then arabinose islands, as I mentioned earlier in my presentation, we consider them as dietary fiber. And these are, what are the dietary fiber? So they have edible carbohydrate, they are edible carbohydrate polymers, which have been They can be obtained from food, or they could be produced as synthetic carbohydrates, but then they are not digested in the small intestine, but they are fermented into the different products in the large intestine by gut microbiome. So arabinose islands, again, we consider them as dietary fiber based on the AACC, American Association of Cereal Chemistry, and they have health benefits. So more specific to arabinose islands and human health, we have done some studies in terms of immunomodulatory properties, but still there is lots of area for research on applications of arabinose islands and how they impact the diet and other human health benefits. So I will be showing you some example studies on immunomodulatory properties very shortly. I realized that maybe I only have 5-10 minutes left. So they also have probiotic properties, and this is a gut associated. So this is how they are. The arabinose islands are, especially in the smaller molecular weight, how they are interacting with the small intestine tissue layers. And because the small intestine, we have epithelial cell barriers, and there are unique receptors that they are interacting with the molecules in the small intestine. So this is one of the model that how arabinose islands can actually interact with the receptors in the small intestine. In terms of immunomodulatory properties, there is some studies related to ferulic acids, and then there are some literature about anti-tumor activity, and then also how they stimulate the antibody-mediated immune responses. And then also they have beneficial effects in patients with impaired glucose tolerance. So we have done some studies using the arabinose island oligosaccharides with different degree of polymerization. So oligosaccharides, they have very small molecular weight. And we know the structure very well. And then also we have done some studies with, these were the commercial samples, but then also I mentioned you earlier, my presentation, we also did some studies by generating structurally tailored arabinose island using combination of different xylanases and arabinopyrimidinases. So in this example, I'm going to only summarize very briefly what were their functions. So arabinose island oligosaccharides with no xylose substitution, they had very limited immunomodulatory properties, but arabinose island oligosaccharides, they had some immunomodulatory properties. Maybe I shall define what immunomodulatory is. Immune system is our defense system, and immunomodulatory compounds, they have impact on the immune system. So we have anti-inflammatory and pro-inflammatory compounds. Which one is bad or good? So it depends on the individual's health conditions. If someone has inflammation that is out of control, we want to control the inflammation, because again, it's part of the defense system, but too much inflammation is sometimes it's not a healthy indication of the immune system. So if the inflammation is out of control, anti-inflammatory compounds are great. But in some individuals, they have immune deficiency and they need compounds that are helping with immune response. And in that case, we call those compounds, we need pro-inflammatory compounds. So that's why immunomodulatory is the compounds, they are pro or anti-inflammatory, and they can actually help body with those immune responses. So based on our studies, oligosaccharides may be more beneficial as gut microbiota substrate, but then we saw that polysaccharides, they had large impact as immunomodulatory responses, immunomodulatory compounds. That's one of the reasons we prepared structurally tailored arabinose island hydrolysates and study their fine chemistry using gas chromatography mass spectrometry. Here the GCMS results, what is the fine structure. And these are, again, what is the terminal xylose. the terminal xylose is shown with orange. If you have unsubstituted xylose, those are with red. And then different arabinoxylane hydrolysates, and then they have variation in the structure. So we had this for 30 compounds, but these are the selected ones that I'm showing you the details. Why? Because they were the most responsive as immunomodulators. And then we measured it using we checked the H here is arabinoxylane hydrolysates, and then we checked that based on the nitric oxide production using the LPS-induced raw macrophages. So, and then we here, again, this is the control. Control had no treatment, but no arabinoxylane hydrolysates, because we wanted to make sure that, and also it was not induced by LPS, because we wanted to make sure that arabinoxylanes are not, or LPS or arabinoxylanes, they don't have any cross interactions in this system. So then we selected some compounds. They had pro-inflammatory activities, but some compounds, they had anti-inflammatory activities. And then we wanted to study those selected compounds first. And also we did some correlation studies in terms of how fine chemistry is impacting those responses. So sometimes negative correlation, and sometimes we had positive correlation. We also did similar studies in the CACO2 and HT29 colon cancer cell lines to see how immunomodulatory effect of arabinoxylane hydrolysates in the LPS-induced TNF-alpha. LPS-induced TNF-alpha production in the cell culture studies. So in summary, I'm going to just go this part very fast. I realized again, there were some strong correlations, negative or positive. There were some strong correlations between the fine structures of arabinoxylanes and then how they actually control the immune response in our body. So why this is important? Again, these were very basic proof of concept studies that fine chemistry is impacting the immune response. So as you all know, NIH, they released a strategic plan. And one of the major items in that strategic plan is food as medicine, and how what we eat can control our health, and can we use food as medicine. So personalized nutrition, precision nutrition. So based on the individual's diet needs, if the person needs anti-inflammatory or pro-inflammatory compounds, we can actually help individuals to develop maybe diet plans working with dietitians. But here we just wanted to show that the fine chemistry of arabinoxylanes, more specific to as being dietary fiber, and then we have them in most of the cereal-based products, how they impact the immune system at very basic level. The big picture, maybe those compounds can be artificially developed and added into the maybe yogurt mixes or cereal mixes. So these are some references that I used, and again, the studies that from my program I presented today. And if you have any questions, I will be more than happy to take it. Thank you.
Thank you very much.
Okay. I wanted to make sure I was done like in 50 minutes. Thank you, Luis.
Okay, thank you. Any questions? ¿Alguno tiene alguna pregunta? Puede escribirla en el chat o hacerla directamente.
Yeah, they can write into the chat box, or you are welcome to translate me if they want to ask in Spanish.
Okay. Any question?
I have a question.
Okay, go ahead, Carolina.
Okay. Thank you so much for your nice presentation. It was really interesting. I have two questions. The first one is, what is the purpose of hydrolyze the leakage of the aranobinophilans?
Yes. So like why we hydrolyze them?
Yeah.
Okay. So let's see. I will show you some slides. So normally, you know, this is the cartoon version of the Arabinose Island structure. So it's beta 1,4 linked, and the structure is different from you know, the fine structure is, it can be different from one variety to another variety. I understand it. We just wanted to know how fine chemistry is impacting biological functionality for Arabinose Islands. So what that means is we know that there is variation in how much monosubstitution you have, how much disubstitution you have. Maybe you have some areas that are naked, there is no substitution. But which one is more beneficial for human health? We do not know that which one is more functional biologically. So we wanted to create structurally tailored Arabinose Island hydrolysates by treating them with different enzymes that they have different functionality. So again, the red color one is the xylanase. It's only hydrolyzing from unsubstitute. We know that. So it is decreasing the molecular weight, but it's just creating that terminal xylose residues with no substitution. So the second xylanase we use, it was hydrolyzing the xylose backbone next to monosubstitute xylose. Once it does that, it is also changing the molecular weight, but also it's creating a xylose hydrolysate that has monosubstitute xylose, terminal xylose. So we had and then we treated we treated the Arabinose Island at maybe different timelines and also in the different order of these enzymes. So we had wide range of structures that they are different in the molecular weight and also fine chemistry in terms of substitution pattern. And then we studied them because again, then we checked biological activity. So then we knew at very basic level what are the most desirable anti-inflammatory Arabinose Island hydrolysates, or what is the most desirable chemistry for pro-inflammatory xylose residues. That's why we designed it, and this is how we created those hydrolysates. And this is published. The more details in terms of how they were prepared, I can send you.
Yes, thank you.
Thank you. So yes.
I have another question. The
Sure.
question is, what are the conditions of the fermentation during baking?
Oh yes, yeah, those are like 85% relative. So they were in the fermentation cabinets, 85% of relative humidity and 38 centigrade.
Okay, thank you for your answer and your time.
Yeah, thank you. So Luis, there are a couple questions in the chat box. In which extent the Arabinose Island affect the viscoelasticity of the dough? Is it mainly the elastic component? Well, it depends. It's viscoelastic. So that one, I think I will, but in which extent they... So Arabinose Islands, they actually have the major impact because of their relation with water and their interactions with protein. They impact the viscoelastic properties of the dough. And in terms of elastic versus the viscose, I would say more in the elastic, but I do not know directly because I have not done rheological studies myself. We collaborated with food engineers. Let's go to the second. When you talk about low, medium, high molecular weight, what molecular weight you mean? Oh, so if it is lower than 50,000 Daltons, kilodaltons, we just say low molecular weight. So if it is in hundreds, thousand, higher 50, 60, 70 to 100,000, maybe medium, and higher than 100K is going to be high molecular weight. Thank you.
Yo tengo una pregunta más. Thank you, Dr. Sinki, for all this information. I have a question about the gut microbiota. The immodulatory property of the arabinoxylan is through the gut microbiota action or is for other physiological paths?
Yes, so the one that we did, they were in vitro cell culture studies. So I did not personally myself work with impact of arabinoxylans on gut. I did not show them in this presentation, but at Purdue we have a professor. His name is Steve Lindemann. I collaborate with him on some other studies. They have not been published yet. He also has his previous publications on him and Dr. Bruce Hemaker. They have publications on impact of arabinoxylans on gut microbiota, lots of studies, but and they have shown that there is significant impact on the gut microbiota community, and then there is also preferential order, which are first used by microorganisms. I do not remember on top of my head, but again, Bruce Hemaker, Dr. Steve Lindemann, they have studies, but I have not shown them today. In terms of my studies, the ones that I show you, they are from cell culture studies in vitro, and then they are mostly in the small intestines.
Oh, thank you, Dr. Simki.
Small intestine studies, yeah. Thank you.
Other question? I had a question, Dr. Sinsek.
Sure.
You mentioned about the interaction between arabinoxylans and ferulic acid. Where is produced those interaction? During milling, during baking?
No, they are actually there during the like how when the grain is developed in the field. It's part of the biosynthesis of arabinoxylans during the grain growth stage. So we do not form those interactions. I mean, they are part of the structure. So some ferulic acids, we can find them in free forms, and some of them are part of the cell wall structure. So they will be part of the cell wall structure, but we are not synthesizing that. They just genetically produced during the grain filling stage. Grain development in the field.
Okay, thank you. Because in Mexico, you know that the maize is nixtamalized before to produce tortillas. And there are studies that show that in the nejayote, that is the wastewater after wash the maize after the cooking under alkaline condition, is produced those interaction between arabinoxylans and ferulic acid. Do you know about of this?
Yes, actually, so arabinoxylans, water unextractable arabinoxylans, they could be high pH alkaline conditions. They actually break down that cross-linking. So they destruct them, they do not create them. alkaline conditions. So then you can actually extract arabinose islands more.
Okay. Okay, thank you. Because it's very interesting because when the interaction arabinose island and ferulic acid reach to the colon, because you know it's not possible to hydrolyze in the small intestine and reach to the colon, that is ferment is the fermentation, but the microbiota. And after the microbiota is to hydrolyze the arabinose island and to release the ferulic acid and to produce and the antioxidant film in the large intestine. I think so that is good for the microbiota, because I don't know if those products of the fermentation, perhaps ferulic acid is to reabsorb and to go in the physiological activities of the body. I don't know. What do you think about?
So are you saying that I'm sure ferulic acid also has impact on all this, and then we just specifically focus on the carbohydrate characterization, but so I also talked about oxidative gelation, and ferulic acids are also important part of the oxidative gelation and cross-linking. But so is your question more specific to the impact of ferulic acid on gut microbiota?
And the antioxidant activity in the large intestine?
I'm sure there might be some studies around it too. So, but I am not familiar. So we have not done such studies with ferulic acid. But
okay.
It will be something interesting.
Okay, thank you. Thank you very much.
I wonder how ferulic acid will directly impact the gut microbiota and which community will be more sensitive.
Okay.
Maybe, it might be one thing that I can check if anyone has published.
Thank you. Other question?
So he's asking, like Mario asked, if there is a product on the market. So there are studies, impact of arabinose islands and immunomodulatory properties, but is there a market in the product? I do not know that there is something specifically marketed as it's going to be immunomodulatory, high in arabinose islands. I have not seen it.
Any questions? Okay, thank you. Thank you very much. Mario, you have the...
Yeah, he already asked, I think.
Thank you, Luis. Thank you.
Thank you so much.
I'm going to show the comprovant of the conference.
The certificate. Yes. Okay.
Oh, okay.
Okay. Thank you very much. The certificate is for the Instituto Politécnico Nacional, Centro de Desarrollo de Productos Bióticos. Award the present certificate to Dr. Senay Simsek, Professor and Head Dance Chair in Food Science, Department of Food Science, Purdue University. For his presentation as a speaker with the lecture entitled Insect in the Arabinohyland Structure and Functionality in Weight, present virtually at the seminars of the Doctorate Program in Development in Biotic Products, Yautepec, Morelos, 4 de abril de 2022. Thank you very much.
Yeah, thank you. It's my pleasure. It's so nice. Thank you. Yeah,
Okay.
Luis. Yes, thank you so much for the opportunity. I hope we can host you again at Purdue University. Thank you, Mario, and thank you everyone for attending. Thank you.
Thank you. Good afternoon.
Thank you very much.
Bye.
Bye.